The purpose of this short lecture is to go through the cell cycle, and we're going to talk about cell division and how cells replicate. And this is primarily via a process known as mitosis. So, the cell cycle in general is just the life cycle of a cell, just as a cell is going through its normal routine throughout its lifespan. It goes through different phases. So, when cells divide, most cells of the body are going to divide via something called a process called mitosis, and our sex cells actually divide by a process known as meiosis, which we are not going to focus on in this section. So the somatic cells would be the most cells of the body which undergo mitosis, whereas the sex cells or gametes undergo meiosis. So that'll be a story for another class. So two major phases of the cell cycle, and this is referring to the somatic cells. So we have interphase and the mitotic phase, or mitosis. So most of the cell's lifespan is spent in what is known as interphase. In an interphase, not a whole lot is going on. So you have three sections of interphase. So you have G1 or gap 1, sometimes referred as the growth 1 phase. Then we have an S phase or synthesis phase, and then we have a second growth phase or gap phase or gap 2. So G1, S, and G2. So in G1, the cell is just doing its thing, just doing its day-to-day processes, and so not a whole lot is going on here. But when we get to the S phase, that's when DNA is actually replicated, and this is in preparation for cell division. So the S phase or synthesis phase, DNA is replicated, and you still get some more growth there. And G2 is just the continued growth phase. So one side note here, some cells are stuck in a G0 phase, and all that means is that not all of the cells in the body divide. So some cells just do not divide, and they never get out of this G1 phase. So they're just said to be in a G0 phase. So there's no division, no DNA replication, etc. But back to the rest of the cells, after this pretty long interphase, G1, S, G2, we go into the mitotic phase, and that's when a somatic cell actually divides and replicates. So for the mitotic phase, there's two major parts. There's mitosis, which is technically the division of the nucleus, and then we have cytokinesis, which is the division of the cytoplasm. And there's definitely some overlap here, but you can see the mitosis phase takes up the bulk of this mitotic phase, and then cytokinesis is relatively rapid towards the end. So there's four phases of mitosis, and in order they are prophase, metaphase, anaphase, and telophase. And then we get that cytokinesis that we referred to, splitting of the cytoplasm. So for prophase, which is the first phase of mitosis, basically the cell is beginning to the things inside the cell, I should say, are starting to break down. So the chromatin, which is the DNA, condenses into organized chromosomes, and you get the formation of two sister chromatids joined at what's known as a centromere. So this gives you an idea of what it looks like. So this, if I point where the pointer is here and here, these are two sister chromatids, and so they're replications of one another. And the reason that happens, because remember in the S phase of interphase, you had the replication of the DNA. So the DNA gets replicated. Now you end up with two copies of the DNA, and they're known as sister chromatids. So this is one sister chromatid, here's another, and then for another chromosome, this would be a sister chromatid, and that would be another sister chromatid. Other things going on, you get the nucleolus breaking down, which is inside the nucleus. And then you start getting what are known as spindle fibers elongating from the centrosomes. So if you look here, we get the centrosome, which consists of the microtubules, and you get another one on this side. And what starts to happen is the microtubules expand a little bit, and you start getting the formation of what's known as the mitotic spindle. And it looks like a little oval-shaped structure. And then in addition to that, the nuclear envelope, which surrounds the nucleus, starts to break down. So there's a lot going on in prophase, but those are the highlights. And through a microscope, it's going to look like this. So it kind of just looks like a normal cell. You can see the cell membrane going around. Here's your nucleus. So nothing overly fancy about it. Just looks like a normal cell. Then we go into the next phase, which is metaphase. And this phase is distinct because the spindle fibers that were coming out from the centrosomes, they attach to the centromere. The centromere is like the center, what's holding the two sister chromatids together. And the spindle fibers or microtubules reach out and attach there and basically force the chromosomes to line up in the middle of the cell. So what you end up with is an organized mitotic spindle that looks like this oval, and the spindle fibers push the chromosomes into the middle and they line up in the middle of the cell. And you get this structure called the mitotic spindle. So through a microscope, this is what it looks like. So this is probably the easiest phase to identify through a microscope. So you see a nice line in the middle of the nucleus. That's all the chromosomes lining up in the middle, and you can kind of see the mitotic spindle formed right there. So relatively easy to identify. So metaphase is marked by the chromosomes lining up in the middle of the cell. The next phase, anaphase, is when the microtubules start to retract a little bit. So you can see that initially these microtubules or spindle fibers stretched out and lined up the chromosomes in the middle during metaphase. Now they're going to retract and basically rip the sister chromatids apart at the centromere, and one sister chromatid is pulled towards one side of the cell, the other is pulled towards the other side of the cell. And this is in preparation for us to get two cells at the end of this. So the cells are going to divide. So again, anaphase, relatively simple. It's just the sister chromatids are pulled apart and dragged towards opposite ends of the cell. And through a microscope, it's going to look kind of like this. So if you can envision these chromosomes kind of being pulled back towards the opposite sides of the cell. Then the last phase, telophase, in general, it's kind of a reversal of prophase, but you start getting a formation of two new cells. So you get two new nuclear envelopes formed around the new nuclei. And you can see the sister chromatids are in here now, and that's going to become the DNA of the new cell. The chromosomes start to uncoil a little bit and go back to the chromatin fibers, and then the mitotic spindle just starts to break down. So through the microscope, it's going to look kind of like this. So it kind of looks like two cells that are fused together because they're just starting to branch away from each other and divide. Then the last part of this is just the splitting of the cytoplasm, and during the endotelophase is when you get this. So cytokinesis, literally splitting of the cytoplasm. So you get this little ring of proteins here, which is called a cleavage furrow, and it just kind of pinches and pinches and pinches until the two cells separate and gives us two new cells, which we'll call daughter cells. So at the end of the day, we had that one cell, it underwent all these steps, and now we end up with two cells, and that's basic cell division.